Welcome everyone. Today we will be studying evolution, and within that topic we'll look at micro, macro, and endosymbiont theory or hypothesis. Just as a quick refresher, this is the geologic timescale. If you've never had Earth and Environmental Science, then we need to look at this much more extensively, and we will study it a little when we get to geologic time. But we start out with Earth's formation, or Earth's formation, right around 4.56. I've heard a couple of different estimates for that, but it's right around 4.5 billion years old. Um, Through most of Earth's history, if we look along this line here, most of these high points we have are geological. We do not see life forming until right around here. These are apex chert fossils. We have the very, very beginnings of life, probably right around 3.8 billion years ago, with the first fossils showing up around 3.5. We have photosynthetic organisms right around 2.5 billion years ago, and then life progresses onto, we have oxygen becoming prevalent in the atmosphere, and multicellular, or cells with nuclei, and then multicellular life right around here. And then the majority of the animals and plants you are familiar with form in the Phanerozoic, with humans showing up right at the very last smidgen of time. The early Earth seas were thought of and referred to as a primordial soup. They contained a cocktail of all the chemicals and simple ingredients that are necessary for life. This is this was the prevalent theory for a very, very long time, and we'll get to some issues with that in a few minutes. Um, the first person to come up with a reasonable hypothesis for the beginning of life was the heterotroph hypothesis by Oparin. Alexander Oparin, in 1936, stated that the earliest organisms on Earth must have been simple heterotrophs, organisms, as we learned, that don't make their own food. He theorized that heterotrophs ate all of the organic molecules around their environment until they didn't have enough food source. At that time, organisms that could make their own energy evolved, so autotrophs were making their own food, and these could have been chemotrophs and photosynthetic organisms. With photosynthetic organisms, we have an increase in oxygen in the environment, and with oxygen came aerobic cellular respiration. Respiration had happened before, but it was only anaerobic. And remember, aerobic respiration is much more efficient than anaerobic respiration. Because of the added energy that organisms were able to make with oxygen available, we develop eukaryotes, which is what you and I are. Stanley Miller was a man working under another gentleman called Harold Urey. The two of them were working at the University of Chicago, and in 1953 proposed an experiment, and this was largely led by Stanley Miller, where methane, ammonia, hydrogen, and water, all of the chemicals that were thought to be in the early atmosphere, were added into an airtight container. It's very important that there was no oxygen present because the early Earth had no oxygen in the atmosphere. The atmosphere was heated, or this container was heated, and then shocked with electrical sparks. This was to simulate lightning. And the attempt was to see if organic molecules could be made from non-organic material, from all these chemicals. In a few weeks, the chemical reactions did produce 13 amino acids and uracil, which is one of the nucleic acids that's found in RNA. These were the building blocks of proteins and DNA and RNA. This was a monumental experiment. The Miller-Urey experiment is its general name, and it was considered for a very long time to be the explanation for the origin of life on Earth if we use the primordial soup model. And this is a diagram of what went on. Again, they heated this chamber with all the gases and water and ammonia. Oh, I'm sorry, up here. Methane, ammonia, water, and gases to simulate the atmosphere. It was shocked. And then down here, it's cooled in a condenser and then reheated. And that the heated gas travels up here. And they could actually collect all of these materials. And that's where they found the chemicals that they were looking for. I believe down here is actually the collection chamber where they would collect all that material. There are a few problems with this experiment. Now, the new data that we have suggests that atmospheric composition of early Earth was actually not what Stanley and Miller thought. That was the composition that people had thought was in the atmosphere for a very long time. Stanley and Miller were following the knowledge that they had available to them. But new data suggests that the atmospheric composition was slightly different. If we repeat that experiment to make organic compounds with the new data, it is very, very hard to get the same organic compounds. It's been largely unsuccessful. So while this is still a huge experimental breakthrough, lots of new ideas have emerged about the foundation of life on Earth. Some of the theories that are coming about are that life began at ocean vents, where all of that heat energy is available. And I've heard some scientists say that all of the organic compounds, amino acids and nucleic acids, were extraterrestrial, that they came from asteroids that have struck the Earth. And we have found some chemicals that are necessary for life in asteroid composition. Probably one of the leading theories right now is that life on Earth began in the soil. Because all of those inorganic compounds are concentrated in the soil, and when you add water and heat to it, it is likely that you will form these organic compounds. So we don't have a very clear-cut decision on where life began. There's a lot of debate among scientists, but you do need to know the Stanley Miller experiment because it was the first true attempt to replicate those conditions. Lynn Margolis was a doctor that came, or a research scientist that was looking at the evolution of eukaryotes. And if you don't remember biology, a prokaryote is a single-celled organism with no nucleus. A eukaryote has a nucleus and membrane-bound organelles. You and I are eukaryotes. She proposed that the evolution of eukaryotes could be explained by endosymbiosis. Endo meaning within. We have endocytosis, which is a process of cell eating, and we've seen cells do this, where they engulf smaller cells, but they usually digest those smaller pieces as food. Endosymbiosis is a theory that says that once those small pieces were engulfed, they were not digested, and instead the two cells lived together in a mutually benefiting relationship. Her hypothesis said that the mitochondria in eukaryotes was the result of an endocytosis event of aerobic bacteria, and that chloroplasts were the endocytosis of a photosynthetic bacteria. In both cases, the large bacteria that encompasses the other two would be an anaerobic bacteria that would not otherwise be able to exist in oxygen. And this would be a mutually beneficial symbiotic relationship. So this is just a general overview of her theory that aerobic bacteria that require oxygen were ingested by anaerobic bacteria, and because of that, both of them were able to survive. I know that's a lot of huge terms. If you click on this video, I'm not going to do it now for time's sake, but click on this video and I'll post a link on the website. This explains endosymbiotic theory in very plain English and is much easier to understand. So do take the time to do that. All right, moving on to evolution. We will look at macroevolution, macro meaning large, versus microevolution, with micro meaning small. Macroevolution is going to result in a new species or speciation. Microevolution results in a change within a single population. This could be, here we have a phylogenetic tree of macroevolution of dinosaurs. And over here I have microevolution, and this example would be an insect that over time becomes resistant to a pesticide. Very, very important. If you remember absolutely nothing about evolution, remember this. Populations evolve. Individuals do not. A person does not evolve. A plant does not evolve. Instead, the entire population or the entire species evolves. You are born with adaptations that encompass evolution or that drive evolution. No one individual is able to evolve on their own. There are several different mechanisms through or that drive evolution. This is how variations within a population become introduced and spread throughout the whole species that will eventually result in full-scale macroevolution. These are random mutation, genetic drift, gene flow, preferential non-random mating, and natural selection. We will probably not get through all of these in this video, but we should be able to cover them in the next one. First of all, mutations. Mutations are mistakes in DNA replication. So when cells divide, they mess up every once in a while. And this happens actually very, very commonly in mitosis. So this is the ultimate source of all genetic variation. Whenever a gene has a mistake in it, it forms a new allele or a new version of that gene. That version may kill the organism, it may give it an advantage, or it may not show up at all. Mistakes or mutations are only passed down to new generations if the mutation happened in a gamete or in a sex cell, so in an egg or a sperm. That's the only time. So most of the mutations that we see are either silent or they lead to disease like cancer. Cancer is a mutation that occurs because of a mutation that controls how often a cell divides. This map shows a picture of malaria occurrences, which are places where malaria is common, that's in pink, and places where sickle cell anemia is common in blue, and where they overlap is going to be in purple. And if you remember from biology, if you have sickle cell anemia, if you have both copies of the defective gene, that is mostly fatal, especially most people with sickle cell anemia do not live past their 20s. With modern medicine, we are able to help people with sickle cell anemia live much longer. But if you have one version of the defective gene for sickle cell anemia, you actually have an enormous amount of protection against malaria. People with one copy of this defective gene are not affected by malaria most of the time. So places where sickle cell anemia is very common tend to be places where malaria is very common, since that gene has been preserved in those populations because it is beneficial, and people with one gene do tend to live longer because they don't get sick. If you click on this link when you have some time, this actually takes you to a very wonderful resource on ecology and evolution. It's a textbook resource that you can read through if you're having any issues understanding some of these terms. Our second driving force is genetic drift, and this one's it's easy to understand, but it gets confusing. It's one of the basic mechanisms of evolution. In in every generation, some individuals just by chance leave behind more descendants than others. And the more descendants you leave, the more genes you leave. So those genes become very prevalent in the population. The difference between genetic drift and other forms of evolution, or other driving factors of evolution, is that genetic drift is by chance. The ones that pass on their genes to the next generation are considered lucky, for lack of a better term. They're not necessarily healthier or better individuals. It happens to all populations, and there's no avoiding it. Sometimes things happen by chance. And the example you see down here is a population of insects where some are brown and some are green, and by chance, some of the green ones get stepped on and killed before they're able to reproduce. That means my new generation will have more brown individuals than green individuals. If this happens over several generations, then the green variety of this insect may cease to exist in this particular population. This is not because the green one doesn't fit or is less competitive, it's just by chance that those happen to be killed more often. I am going to stop there and pick up with our next video to cover the remaining parts of evolution.